my talk will basically be an extension of Danny Schanz's previous talk, because he raised important questions, and in our interaction with him and looking at those challenging samples that he provided, we learned a lot about how to improve our software, how to expand like the possibilities and maximize the utility of this single assay that has the potential to replace so many other assays in the laboratory. Let me first, reiterate what Danny already said. How are we still using HC38? There's a better alternative available. The second topic will address one of the big feature requests of Danny, the methylation analysis. I'm going to present you results also from the data that Danny provided. And I will also talk about a new feature that will be introduced with our new software release in a few weeks regarding this specific analysis of paralogs genes. T2T has been around a couple of years. It adds hundreds of megabases of sequence information to the reference genome. I just want to remind you, it's the sequence that has been missing in HC38 are particularly those P terminals in chromosome 13, 14, 15, 21, and 22. And every time that a structural variant is linked to those regions, we will not be able to describe them properly in a setting that references HC38. So from a technical point of view, there are many reasons to switch to T2T, but of course, kind of the most important question is, who is going to fund a new GNOMAD version 5.0 for the T2T genome? Because without this resource, I think it doesn't make sense to introduce the new reference genome to clinical practice, because it will just there will just be so many variants that we cannot interpret. Let me move on to a more positive note, methylation analysis. This is a very exciting topic also for us, because it provides so many opportunities to give clinicians more relevant information about the patient. In our first version of methylation analysis in the varvis software, we focus on clinical utility. That means on established differentially methylated regions in imprinting disorders, for example. I'm going to show you data from nanopore whole genome sequencing and also adaptive sampling, exclusively PCR-free assays, and some of them provided courtesy of Danny Schanze and his team. I'd like to remind you, methylation data analysis from long read is well established. There's, for example, this recent study across a large number of individuals where methylation data from long read sequencing was compared, for example, to the standard of care of methylation arrays. So there's no question about analytical performance of this technology. I'm simply going to present to you data from Oxford nanopore sequencing. The data that you can obtain from long read sequencing is very rich in details. You can obtain information very nicely resolved across the genomic coordinates. So this is an example here of the locus SNRPN, promoter region of this gene relevant for Prader-Willi syndrome, for example. And you can see here two curves, a green one and a blue one. The blue one is one allele, the green one is the other, the haplotype number 2. And you see that the blue one exhibits high methylation. On the y-axis, I'm showing you percent of methylation. X-axis is a genomic coordinate. So one haplotype, one allele is highly methylated, the other one is basically not methylated. That's what you would expect in an unaffected individual. The maternal allele is methylated and deactivated, and the paternal allele is actually expressed. So everything as we expected to see. This is simply a different view on the same data, averaging methylation across the region that I showed you before. And as you can see, both in the loci MAGEL2 and SNPRN, we have high methylation in one allele and basically no methylation in the other. So the data is of high quality, it's very low noise, and you can make very clear distinctions between alleles in this case. As Danny highlighted, the quality of your long read sequencing results may depend on DNA quality, on fragment size, on capacity of your flow cells. So you may not be able to properly phase, to properly assign all data to these two haplotypes in these regions. So we may have data that is unphased. And you need to have, when you're assessing these cases, you need to have information about that too. What fraction of your data is unphased? What's the state of methylation here? And as you can see here, I added a bar chart for those unphased data points. And as you can see, as expected, it's about 50% methylation, which you would expect if you mix the two alleles in an undiscriminatory manner. So this is just a brief check of an unaffected individual. Now, if I want to look at a larger cohort of patients, I would like to do this in a very fast way. When you want to screen for imprinting disorders, I'm particularly a fan of good visualization. So I like heat maps, false color maps of data. You can see here on the y-axis, there are the 3 loci that I showed you before, the ones that are relevant for Prader-Willi syndrome. And I'm showing you data of 9 whole genome samples provided by Denny Schanze. And you can see immediately that 3 samples are standing out here. So the yellow level, 50% methylation level, that's the average between the two differentially methylated alleles. That's unaffected. And you can see here that sample 4, for example, has a very high level of methylation. And let's take a closer look at this sample, what it looks like in a detailed view. You can see here clearly both haplotypes, the green and the blue curve, follow a very highly methylated profile. So it's very clear MAGEL2 is methylated. The paternal allele is basically missing. And just for comparison, this was what it looked like in the unaffected individual. And also the other locus, as in RPN, is clearly highly methylated. So you have both very high resolution across the loci and very low signal-to-noise ratio. And also when looking at the bar chart, that gives you kind of the average across these regions. This is exactly what we expected from the previous charts. We also compared this in all these samples with the gold standard, at least the gold standard in Denny's laboratory, MLPA, methylation-sensitive MLPA analysis, and the concordance is very high. So there's no doubt this assay is both sensitive and very accurate. I'd like to show you data from another cohort as well, data that we were happy to analyze together with the University Clinic of Leipzig. Data is courtesy of Danny Popp there. So these are 16 whole genome samples that we also analyzed in these specific loci. You can see here now, of course, these were pathogenic cases. There was abnormal methylation in all of them, but you can see here in this false color chart that at least 2 of them are kind of standing out because their level of methylation is not close to 0, but it's a little higher. Let's take a look at sample number 12, what the reason is for this different level of methylation. And when we're looking at methylation, for example, in MAGEL2, we can clearly see, okay, one of these alleles, the green allele, is very close to 0% methylation, but the other one has retained some methylation, or it's probably a mosaic case of methylation. So this highlights this method is very sensitive and can probably also distinguish between different levels of methylation. So far, our analysis is now focused on on the use cases with very clear clinical utility. Imprinting disorders, as I said, you may have seen in the report, you can basically click through a number of groups of genes, of loci that are related to specific diseases. We are also performing the methylation analysis on repeat expansion loci. Some of them are known to, the methylation is known to impact pathogenicity in some loci. And we can also do the same analysis across tumor suppressor repair gene promoters. If you know of other applications that are clinically relevant, that are of interest to us, to your clinical work, please, I would be happy to have a discussion about this with you, because we are learning so much. We're gathering so much information now about, where this information would also be interesting and how you would want to analyze this data. This is what we're currently focusing on. Now to my third part of my talk, the specific analysis of paralogous genes. You may be asking yourself, why do we need another algorithm, another tool to analyze long-read sequencing data? Aren't they supposed to be perfect, those long reads? Well, fact is, even if your reads are very long, there are still regions in the genome that are so similar that an alignment algorithm cannot really accurately decide which place this read belongs to, simply based on sequence similarity. You also need to take into account the entire context of the haplotype, the entire phasing information. And that's just not kind of the standard way to analyze this data. And the result is that there may still be some reads assigned to the paralog that should actually be assigned to the gene. And to overcome this limitation, we apply an additional step of analysis for these known loci, where we extract the reads that were assigned to gene and paralog, or gene and pseudogene, and we force mapping to one locus, to one gene, and then perform the phasing and separate the individual haplotypes. And this gives you a very clear overview of reads that can be clearly assigned to the gene, and reads that can be assigned to this other locus that is similar but not 100% similar. And also this method has been evaluated on larger patient cohorts, and both sensitivity and precision have been demonstrated, again, by the group at Radboud University Clinic. And there's also a talk tomorrow about this topic on Sunday at about at 11 o'clock. So the algorithm is very powerful, but the output is very complex still, right? So you have the raw output is probably not digestible for a human, or it would take a very long time. So we designed a suitable and simple user interface to interpret the data. So what you get in our software is on top a view of all the variants that were found in this complex locus. And below that, you have a graphical representation, which haplotypes were identified in this locus. And let's take a look at a few examples, what this looks like in detail. So here, this is a very simple example, GBA1 and its pseudogene. And in this graphical representation, each column represents a SNP, a single nucleotide variant. And green means the SNP is equal to the wild type in the gene. Orange means it's deviating from the gene sequence. So we have, and each row is a haplotype. So we have two haplotypes that are green, that are very similar to the gene sequence, and two haplotypes that are differing from the gene sequence. And we're highlighting here just the differences, right? That's why the picture is so clear. If you would look at the raw alignment, you wouldn't notice the differences so much. So you have those four haplotypes, and all the variants that you saw in your table above are annotated. In which haplotype have they been found? So you can easily filter for variants that are found in the functional genes, and this facilitates classification of those variants a lot. This is a simple case, just with the GBA1. A more complex locus is the RCCX box, where you don't only have very similar genes just being part of the genome, but you can also have recombination events between gene and pseudogene. So in this case, we have one haplotype that is fairly green, which is coinciding with the functional gene. So we have only one copy of the functional gene, and we have one chimera A2B1. And again, this is annotated. Your variants are annotated with this information, and this facilitates the classification of the variants because you have all the annotations about the functional consequence of the variants. Here another example, again, from the RCCX box. And again, you can see we have one haplotype that is fairly green, which is the functional gene. We still have only one copy in this case. And we have another chimera, but now B1A2. And again, this is something that you see at a glance, and where there's no ambiguity, or there's little ambiguity, and you can quickly identify how you should interpret the variants that you found. And so with this is kind of a preview of software that's going to be released in a few weeks. You can have a look at it also at our booth if you're interested. And with this, I'd like to close my talk, and we can move on and basically take questions.